Hello, my name is Jack Delli-Santi. I was selected to receive the Conservationist Award last spring at the 2019 Texas State Science and Engineering Fair. I conducted an experiment testing whether carboxymethylcellulose could be used as a remediation strategy against zebra mussels. Zebra mussels, which are a highly invasive species of bivalve, have a uniquely evolved gill structure which allows them to siphon particles out of the water down to 1 micrometer in length. This also renders them extremely vulnerable to hypoxia, meaning they can be killed off due to a lack of oxygen. Current remediation strategies, like biological pesticides, can be harmful to other species in the environment. Methods like the swish and spit technique rely too heavily on independent boaters to clean out their hulls, or chemical bombardment, which affects only a small portion of the population, can be ineffective. Carboxymethylcellulose, or CMC, is a common food additive and is used as a thickening agent. CMC is generally recognized as safe by the FDA and is also biodegradable and biologically inert, meaning it's a perfect candidate to be used in an ecosystem where you cannot regulate where things flow to. CMC creates hydrogels when deposited into water and is also extremely cheap, making CMC a cheap solution for dealing with large populations of zebra mussels. My experiment, testing 3 different CMC concentrations, tested the effects of CMC in 3 different environments in which zebra mussels are commonly found. My acute exposure experiment replicated a riverbed where CMC would be deposited and gradually swept away by the current. Mussels were exposed to CMC for 30 minutes before being placed back into their standard pond water. To replicate a lake ecosystem where CMC would be deposited and gradually diluted, I designed a prolonged exposure experiment, exposing mussels to CMC for 48 hours. In an attempt to replicate a congested pipeline, I designed a chronic exposure experiment. This experiment exposed the mussels to CMC for 6 days without any dilution. During the 30 minutes of acute exposure, there were notable changes in mussel siphoning activity and behavior. However, after 48 hours of recovery, there were no long-term changes. After the 2 days of prolonged exposure, while no mussels had died, there was a significant drop in mussel siphoning activity and extremely abnormal behavior. Across the 6 days of chronic exposure, mussel siphoning activity dropped drastically after 1 day and remained low over the next 5 days, while the number of diseased mussels continued to rise across the entire experiment, peaking at just over 50% mortality. While further testing of this technique needs to be conducted in situ, I believe this method can be best employed in confined congested areas such as the pipeline replicated in the chronic exposure experiment. Due to CMC's nature, it could also be used in replacement of agar or carrageenan gel as a culture for growing mussel toxic bacteria. Thank you to Lynn Seaman for reaching out to me and inviting me to share my work, and to all of you for your work in conservation. I'm sorry I could not be there with y'all for this amazing event. I'm currently in Florida at the University of Miami studying marine science and biology. Last spring, I had the amazing honor of competing at the Intel International Science and Engineering Fair in Phoenix, Arizona, where I placed 2nd division in the world in Earth and Environmental Sciences on this project. ISEP was a truly life-changing event, and I would like to thank all of y'all for your support of my experiment. Thank you so much.